So thank you for attending this last session of the workshop. Yeah, some of you seems to be tired already. So the idea of this last session is to present the summary of the 4 breaking sessions, and so questions, ideas, and the short summary, and to discuss about the next steps. So we have 1 hour and a half. So the proposal is to have approximately 15 minutes per group for the presentation. And we are going to begin with group A, B, C, and D. And after that, we're going to have a general discussion, and we have to keep 15 minutes for presentation of what are the next steps. Okay, so I propose we begin right now. And for people online, don't hesitate to participate. So you can raise your hand, and you can speak also. So don't hesitate to participate. So you're the rapporteur for Group A. So Pike, you're welcome.
Thank you. And I have a few slides that should be there. Yes. So Group A dealt with the prenatal exposure. We were mostly on site, but had a few online participants. And I think we were in by far the warmest room. So it was pretty intense, and sometimes it's terminology of pressure cooker is used. Ours felt like that. Could I have the next slide, please? Oh, here. Nope. Nope. Is this the right one? It does have a pointer. Oh, yeah. There's a lot we discussed that is not in this summary. So if anyone is interested in all the details, we have written notes, and we're not going to go into all the details. But what I'll be presenting here is what we think were the most important things we discussed. As a first remark, we were considering prenatal exposure. So it was only the children and not or only the parents and not the children and grandchildren that were exposed. So in some of the discussions about which generation is which, that was an issue. In our case, it was simple. It was just the parents. Part of what we did was look at what is the important progress made since 2007, so since the last ICRP report on the issue. And we distinguished epidemiology and epigenetics. And in epidemiology, we think an important change is the reanalysis of the ABOM survivor data, which give a consistent, if weak, indication for effects on malformations and pregnancy outcomes, but not so much for germ cells. So it's a fairly consistent picture, even if not significant. And because the issue then is, should we take that into account in risk estimates or not? Another important development is the TRIO studies that are being carried out in several settings, which are giving very interesting results on transgenerational effects. But an issue there is that in many cases, the dosimetry is very limited. So if you're trying to develop risk estimates, they are, until now, mostly of limited use. They do give insight into the mechanisms and outcomes involved, but are not yet so useful for specific risk estimates. Then epigenetics, the consensus in our group was that it's a very young and complex field of research, and the contribution to effects in future generations at the moment remains very uncertain. It's, in our feeling, it's too early for risk estimates in future generations, but it is a potentially very relevant field, so we should continue to explore this, and in particular look beyond radiation research, because this is not just relevant for us. And since we're just beginning to learn here, it's important to, for example, look at people who are working in, say, aging research. Just as a side remark, there's also a lot of new tools, because it's right now, I think things as whole genome studies are fairly standard, but in 2007, those were just emerging. So there's been a lot of technical development since ICRP 103. A brief thing about terminology, because there was some confusion about what should be the F0, F1, F2. And in our group, we had the idea that this is terminology that was traditionally designed for a setting without exposure. So you start with an F0 that has a certain genetic makeup and see how that transmits to future generations without involving exposure. So it's not something that's tailored for situations where exposure does play a role. It doesn't mean it can't be used, but you should just on a case-by-case define what you mean by F0, F1, F2. And there is no one-size-fits-all solution for this. But it's just, as a general remark for the cross-group discussions, just define what you mean by F0 to F2 or even more. Then, we went over all the questions and we have ideas about them all, but I'm not going to go over them because I think that will be just too much for now, because these are the most important knowledge gaps or issues that we identified. We didn't want to call these recommendations because that would be too strong, but these are issues that we feel could be important. As a first point, we think it's important to continue with the whole genome studies, both in the trio studies of parents and children and in animal experiments. And in mice experiments, we've heard on the first day that there are indications that transferring results from mice to humans could be difficult because mice appear to be more sensitive. However, those studies were done in single loci, which could be more sensitive than the genome as a whole. So to get more insights into transferability, whole genome studies could play an important role. Similarly for the trio studies. So there's no specific ordering in these points, but they somewhat go from exposure to outcomes. Dosimetry is an important issue. As remarked before, in the trio studies, there's often very limited information. Those studies right now are often conducted years after exposure, and it's hard to reconstruct what the exposure was. So any new studies should take dosimetry into account, which, well, is probably fairly standard for radiation research, but important here because if you're trying to get to risk estimates, that's really what's lacking at the moment. Then also on the exposure level, variations in dose rates of acute versus chronic would be important, internal as well as external exposure, radiation quality, and this applies to the trio studies, which are now sometimes fairly uniform, as well as to animal studies and to, if possible, general epidemiology. We do not think that either fathers or mothers should be studied more. Both are equally important. Well, no, both could make a contribution, and we do not know at the moment if one is more important than the other. So we should study both. And in human studies, we think ideally they should include 3 generations. So the exposed parent, the child, and the grandchild. This is still in a prenatal setting, because that way you would get the most information about effects in future generations while still dealing with a somewhat manageable lifespan issue. More than 3 generations probably is going to be hard to get all 4 generations alive, or it would require enormously long-running projects. Most existing studies are on occupational and environmental exposure. It's our feeling that it's valuable as well to look at cancer patients, specifically childhood cancer patients, even though they are not very similar to the population as a whole, but they are a pretty large group, and knowing what the perspective for their offspring is important as well. So they're an important group in themselves, plus they might give valuable insights into why some people develop cancer and others don't. One of the important issues that we were talking about was what are the sensitive stages in oogenesis and spermatogenesis? So if someone is exposed, and could you give them any advice about how long to delay a pregnancy? The current data don't give much information about this because they usually start at quite long after exposure. So the time window in which you would be able to say anything about this had long passed. So that would be something that's clearly a gap in our knowledge. And we put together a long list of confounding factors, including lifestyle factors such as diet, which might be ethically more difficult to address because it might be seen as more invasive into people's life when you want to include those into a study. Of course, background rates are important as well. It might be interesting to consider put together quality guidelines to take those into account in a consistent fashion across studies. Another issue we discussed was which outcomes should we consider? Just the ones that we know are relevant for detriment, or the ones that we would like to incorporate in our risk estimates, or any changes? And the consensus in our group was it should be any changes, because for some of those we just don't know if they're going to be relevant for detrimental outcomes. So consider all outcomes, including those with uncertain relevance for health outcomes. Dominic, we did think about doubling those, but in the 3 days that we had here, we couldn't say anything sensible about that. So that's something you'll have to wait for in the future. I hope that when we continue to work on the review papers, we'll be able to say something about that. But unfortunately, nothing for now. Sorry. As already said on the first slide, for epigenetic effects, we think it's valuable to collaborate with people from outside radiation research who are working on similar topics. Transferability from animals to humans could be an issue. However, if we have a thorough understanding of the mechanisms relevant in animals and possibly also in humans as well, then to derive risk estimates from animal data, possibly combined with limited human data, might be helped by using mathematical models, which I think mathematicians could play a role there, and as well in the big data analysis of the large volumes of data that come out of possible whole genome studies. And then finally, all of this, when we were talking about this, it all led to fairly unfocused ideas about what we could study. So as a final recommendation, when developing studies, make sure you have a carefully phrased hypothesis or research question and not just some arbitrary thing, because that will make the results way more interpretable. I don't know if there are any questions at this moment. I think there's still quite a few people from the group here, so they can help me if I don't know the answers.
I think it should be preconceptional exposure. Yeah, it's not prenatal. Prenatal is different. So before you ask, I wanted to clarify.
Sorry, it's just a blind spot in my, I didn't see it anymore.
Everyone, when you're speaking, say your name, where you're from. Thank you.
Okay, we go officially to Rafi from SCK Belgium. So, yeah, I think this is a bit confusing when we speak about F0, F1, F2. You know, for all geneticists, we have only one Bible, is the Mendelian genetics. I'm sorry, but we should not be confusing that. So if you have a male, sorry to go to the basics. If you have a male yellow and a female green, you made them, you do get an F1. This is F0. You get an F1, which is orange. The F1, you meet the children in between. You meet the children and you get the F2, where you find the dominant, the non-dominant. This is only one genetics, and we should not, let's say, start to make confusion of this. I'm sorry, but this is something to be clear for everyone. This is the Mendelian genetics, and we should not say, yeah, F2 or 3 or 3. So this should be clear from the beginning, and we should fit to this, let's say, basics. And I think, yeah, now we are not far. So I agree with all, but just I think we have to be more consistent with Mendelian genetics.
I don't know if you're not already too late, because I've seen it used a lot in presentations so far, but you have a point. Yes, no, this is it. And apologies for saying prenatal when I meant preconceptional, because that's just time for coffee. Yeah, yeah.
So Dominic speaking, it's written 3 generations in humans. Does it mean that you are interested in only 3 generations? You think that after 3 generations, that's enough? Or it's just that you didn't know and you wrote 3 just in case it was the good answer? Why 3? And well.
I think an important motivation there was that we thought that 3 was still practically manageable, and more than 3 for practical purposes becomes very difficult. So it's more a practical issue than scientific.
But so should we recommend the radiation protection system to move from 2, which is considered at the time being, to 3, or is it there's no relationship? It's just a practical aspect for research.
And when we set 3, this was more when we're trying to understand the fundamental mechanisms, look beyond 2 to see if there is anything in 3.
Sorry to complete, but when we were together yesterday, there was a scientific sound to tell you that, because if you address preconception, it's only the gamete. So in the F0, you irradiate the F1, and then at the F2, you don't have any direct irradiation. So 3 generations is enough for preconception studies.
Yeah, that's true. So the 3rd generation has no exposure whatsoever.
But it has to be understood as that's the minimum that is needed if we want to understand the mechanism. Not that's what is needed if we want to have an idea of the whole picture. That's it. So come on, additional questions. You have questions. Don't hesitate. That's no. So I have another one. You said epigenetics, it's too early for risk assessment. Okay, but anyway, at the time being, or next years, do you think we can say that we are not able to quantify it, but it's in addition to what is considered at the time being, or do we think it's considered in what is considered in the radiation protection system already? I'm sorry, I always have questions that are related to radiation protection, but.
So the others in the group, please give extra information if you feel necessary. But I think we had the feeling that it might to a large extent already be included. So there could be health outcomes that we have observed that are influenced by epigenetics, only we don't know it, but it could already be included in the system of radiation protection. But that's a slightly hand-waving argument. We have no solid evidence for that.
I'm sorry I come back to the 3 generations. I am, so yeah, maybe again, coming back to the Mendelian genetics, 3 generation is the way to be sure of transmission of a mutation or a criterion from the parent to the grandchildren. So as I say, from F0, F1, you will have a mix. You cannot see what is what. And in the F2, only there you can see if a dominant or recessive character is appearing, is only in the F2. So I think that should be explained that way, because this is the way we learn it from Mendel. You agree? Okay.
Okay, then.
One line of conclusion for Group A?
Yeah, I'm afraid that will be the very standard, more research needed. No, no, but yeah. So I think okay. So for the risk estimates, the epidemiology does have some new results that we think should be taken into account for quantitative estimates.
Yeah, just Rishi Wakeford, just to fill in the time there, Dominique. We also have, since over the past 20 years, the offspring of the cancer survivors, and they're going to be increasingly important, because childhood, adolescent cancer has a high rate of cure these days. So, and they all go on to have children. So, there are a number of groups, the Nordic countries in particular, where they have these these very big databases that they can link together. Very envious for these things. So they are an increasingly important piece of information on this. So I just to emphasize that point, I think.
Yes, and there may well be ongoing studies that are informative even during this lifetime of the TG. For example, after the session with Europa, I was contacted by a Japanese scientist saying that they are carrying out low dose rate exposures on mice and planning to do whole genome analysis of mutations. So
To check, there are no questions on the web at the moment, so we're not missing those. I think that's the time for me to rush off to the airport then. I wish you a very successful end of the meeting. Thanks.
Thank you, Fike. So you're going to miss.
Oh, yeah. Klaus, did you have a question?
Yeah, I just warn against these childhood cancer survivor studies. Childhood cancer receives inevitably a very complex mixture of different genotoxic agents called cytostatic drugs. The doses are so individual that it would be impossible to derive any estimate on the risk of the additional radiotherapy. This applies even to direct cancer induction, but it would certainly be much more complex and difficult to interpret with regard to genetic risk estimates. So, of course, it would be good to do the studies, but I warn from straight and direct conclusions.
Evgenia, do you want to say anything about the TRIO studies?
No.
Thank you, Professor Trott, for your...
I'm very skeptical about the TRIO studies, but anyhow, not here, not at this moment.
It's very well noted, your opinion. And I'm Evgenia Straumova. And when we discussed in our group and what we agreed to do, we will start with the review of the already existing study and also to see the limitations and strengths of the studies already done on offspring born to childhood cancer survivors. And that also will help us to how the best way is to address the knowledge gap about the transgeneration effects. But first of all, of course, the very thorough, very detailed review of the existing bulk of evidence or knowledge that we will start with.
Okay, so now you can take your flight. Yeah. Thank you again, Fike. So if there is no more discussion on Group A, we propose we move to Group B.
Thank you. Yeah, also before doing the systematical reviews, this meeting, it was like kind of brainstorming, no? Just trying to have ideas on what we have to pay. So we hope that our presentation appears. So we are lucky group because we have two reporters, Ludmila Lutsko and Emily.
I'm Emily.
So, and here is nice group. Also, you can see on the picture. We are mentees to the task group 1, 2, 1. Yes, thank you. So here we prepared a summary of our work, and you can see our work on the picture. Yes, next slide, please. Okay. So before we had different short topical presentations of the group B, and here you can see the topics. Kimberly gave us talk about fundamental biology of embryonic and fetal development, highlighting periods that are sensitive for radiation effects, developmental and cancer induction. Stefan Grisson gave talk about fetal development and radiation, a quick look relevant for multi-generational and transgenerational effect studies. Myself, Ludmila Lutsko, reported about relationship of thyroid hormone levels with exposure doses and intellectual coefficient in exposed in utero by the Chernobyl accidents versus control, as per Belarusian studies. Rafi gave presentations on radiation exposure during brain development and long-lasting persistent effects. Klaus, who is online, very actively participated in our work also today and all the days, talk about factors influencing radiation effects pre- and post-conceptual exposures, and Eric Grant gave talk on advantages and limitations on IERF's data for analysis of radiation effects on offspring and next generations. Actually, some of these summaries we prepared here for each talk. I will just throw in through, and you can just have a look, like a kind of flashes, summaries of results on important points were extracted from presentations. But some of them are not published, so just not to take pictures, or if you take pictures, do not distribute it. Here it was from the first presentations of Kimberly. This is from mine, and some results you can see here on IQ, first on thyroid, and main conclusions. And Rafi, if meantime somebody can wish to comment something the most important, you can do also, but I am going just showing you slides, okay? Of those presentations as a summary, but if you catch attention and you have interest, you can ask and talk to our presenters. Next was by Eric. So the main questions he put on the memory, here to study. It was before from Stefan, and now it's Eric's presentations, and from Klaus. It's very fast. It's a little bit slow. Yeah, there's a lot of text and it will slow. Mm-hmm. Yeah, next. Yeah. Okay, next. Here was nice graphs about 25 cases he explained, I remember, of the bicrosses. And here on those effects in utero doses, depending on doses. And questions to reflect and to follow on. Okay, and then next part, I will give Emily.
So here's some questions from Group D to Group B about post-conceptional exposures. So is the all or nothing rule this still valid for the pre-implantation phase? So it raised a discussion about the deterministic effect and stochastic effect, and as Kimberly well explained, it was more like just a simplification, and now we know that we have to consider more than just a single cell exposure, and more like multi-cell effects, and it also can have effects on the next generation. So we think we could reconsider that. Is there evidence for stochastic induction of malformation during the pre-implantation period? So studies show that there is genomic instability of surviving individuals, and so still a valid hypothesis, but we still have to research on this topic. Can threshold doses for malformation observed for animals be reasonably transferred to humans? So we cannot transfer directly animal data to human because time factor is very important to consider. But we know that animal studies are relevant when we develop, for example, drugs and so on. So according to Rafi, yes, we can consider we need to find a way to translate these results, but they are relevant. Mental retardation, how sure are we with the threshold at 200 milligrays? So the answer is we should update this threshold. Is there new information on deterministic effects after in utero exposure? So yes, there exists data, and we need to check the studies again. Is there enough scientific information available to integrate the risk of cancer after in utero exposure in the calculation of the detriment? So still, we have scientific information on that. The question is how to include them. How can we include this on dose in calculations as the detriment? This is the biggest question. And is there a baseline instant data on congenital malformation available today for different populations? Yes, we have, and Kimberly addressed the CDC study, which is a pretty robust data on congenital malformation before 1 year old. So we should consider that. Some highlights. So these are some highlights, like statements, but also more questions. So yeah, challenges as well, yes. So the first, one of the most important statements was the lack of studies on occupational exposed women. Also, the chromosomal translocations in animals and human studies, something we have to consider to study. The uniform risk of childhood cancer, why is it? Of course, except for bone tumor risk. The importance of non-cancer effects and their role in epigenetics, because sometimes we speak too much about cancer, but we also have non-cancer effects. It's linked with the next point, which is the holistic model approach, so considering the social trauma and other confounders to the effects. Are we missing some health effects that can also impact on the well-being, but not really related to mortality, and we need also to consider. We also address importance to the comparison between the results of planned, like medical exposure, and the accidental exposures. How important are the effects of development in the central nervous system, and which are the sensitive periods? Do we have new analytical methods, and should we reanalyze those old studies with these new analytical models? Because we have some consistent data, not just in humans, but also in animals, and they are good. So should we reanalyze them with these new methods we have? Also, the impact of fractionated dose, once we know that the response in cells are different from single exposure and fractionated exposures, So this is something we should also take into account. The exposure of women to environmental radionuclides. Regarding human and mice, the neurotope defects and how we differentiate that and how we study, how to compare. Some studies they have in the Oxford studies about cancer and how it is in a great order of magnitude compared to the other studies. So should we reanalyze them? Regarding uterine dose and fetal dose, how to relate them. We're doing that, how to relate them. Some studies in mice about the neurological effects, but also the metabolic changes that were found. So this is something we should study in humans. And the biggest question, is there a threshold for IQ effects for irradiation? This is something we cannot, we don't have really a conclusion. And the last one about genetics susceptibility to cancer, considering tumor suppressor and gene deficiency. Some challenges, considering the sex differences, especially from the A-bomb studies. Is it and why the third trimester more sensitive in children? Some registries for CT scans, something we also discussed, the importance of having the registries of medical exposure so that we can then compare and study the effects. And we don't have any studies so far on case control studies from different injuries. So if somebody crashed a car and have to do an X-ray in the arm, and how we compare the exposure to X-ray because of an arm X-ray and not a pelvis X-ray. And this is...
Okay, and this is the last slide to show us our working, you know, our people here online also, and this we are, and our online contributors. Okay, thank you very much. So if you have questions, then somebody from group will reply. Okay.
This is, yeah. Okay. This is Evgenia Straumova, and I hope I didn't miss it through your extensive list of scientific issues you included. But what about not only external but internal exposure to the fetus, especially in the exposure situations when there are some radionuclides that accumulated in certain organs of the fetus? And what I mean specifically, it's the in utero exposure in Ukraine and Belarus, where the exposure took place with radioiodine penetrating the maternal placenta and accumulating in the fetal, fetal thyroid gland. And it's already the result that can also be important for consideration and inclusion into the,
No, it's a great comment, and it's very good. We haven't discussed it on it in detail, but we were speaking about other founders and other internal factors of lifestyle and everything, you know, just in general. But what you're speaking, yeah, I know, I understand that what you're speaking about transmissions is like this. Yeah, it's an important issue. We should search for these studies and in review. Cheyenne, please, your question.
I just had a quick question about something on a slide. If you don't mind going back to the slide that says, is there enough scientific information available to integrate risk of cancer after in utero?
Which slide, do you remember which slide number is?
I don't have the number. I think you just passed it.
You can stop me when I go there, no?
It was one of the later slides from the summary.
From questions?
Yeah.
So questions to group. So first note.
Not this one. I think it's the next one, or one more.
One more.
Yeah. So can you explain what the bolded statement means? Because I'm a little confused about the wording.
Which one? I didn't catch.
It says pregnant women should not be never exposed to IR.
Yeah, it was Klaus who was saying. So it was like the question we were discussing if we should include pregnant women irradiated in the whole population, like exposed population, the whole population. But maybe Klaus, you could reply. Maybe Kimberly. Okay.
Yeah, this is Kimberly Applegate. I'm part of Group B from the ICRP Committee 3. That's really a little bit misleading, Cheyenne, as you may be thinking. Pregnant women are exposed to medical ionizing radiation in rare circumstances because of various situations where they have to be imaged for life-saving reasons or potentially critical situations like when they've had motor vehicle trauma. So should not, never is not quite correct. So I apologize for that. And so they are exposed in certain situations, like life-saving trauma. They get a CT, they get embolization of vascular bleeding, they get sometimes perfusion scans for pulmonary emboli. There are many situations like that. And then 1 in 1,000 women get cancer and have to have workups. And possible radiotherapy. Most commonly it's breast cancer, but it's also lymphoma, melanoma, and a few other cancers. In terms of occupations, there are many, 80% of the world's radiographers and technologists are women. So many of them are in childbearing age, and they, in many countries, including in the United States, they are allowed to practice their profession while pregnant. They declare their pregnancy and are given a special badge. So their exposure is supposed to be almost nothing, but there's it says not involved in occupations related to ionizing radiation. That's not correct as well. So that needs to be, obviously we'll amend that. So does that answer your question, Cheyenne?
Yeah, that makes a lot more sense. It was just the wording was threw me
the
off.
wording is a little bit misleading, but yeah.
Okay, thank you.
Yeah, the statement was like desirable, but not reality, but not really. Yeah. Yeah. We will. Yeah. Okay. Thank you for this. Yeah. Okay. So we will. Yeah. Correct. Thank you, Cheyenne, for your comments, and Kimberly for comments on in. Any more questions? Or suggestions or discussions? Oh, yeah, we have Sarah.
Hi, good morning, and here in the US, I'm Sarah Dumit, virtual attendance here from Los Alamos National Laboratory. Ludmila, thank you very much for the presentation. I have a quick clarification. You had a slide. I think if you move forward, let me see if I can find.
For questions or for highlights?
Yes, keep going forward. You had, I think, yeah, forward.
I think it's the last highlight.
Yeah, it was toward the final slides. It's just a quick.
But it's final because this is final one. I move back.
When you mentioned about the questions, maybe the review about NCRP 175 report. That's the first. Yes, this one.
This one.
Thank you. Yes, you found it. So the second bullet point, You mentioned about the lack of data studies on occupational exposed women. So the clarification I would like to ask is if this occupation is about medical or about nuclear sites.
It could be both, no? Could be both, because I will give to Kimberly.
Yeah, I think, Sarah, you're going to probably raise the NCI studies from the radiation branch, right, that have looked at radiographers, technologists, and physicians. So they have followed within the U.S. Risks, cancer risk and cataract risks. So there are occupationally exposed women and their risk of different diseases, correct? Is that what you're trying to say?
Yeah, because
Yeah,
the
so there are
report,
studies.
yeah.
We didn't go into it in any detail, but we certainly would include them.
Okay, that clarifies the question. Okay, thank you
Right.
very much.
Thank you.
Thank you. Any more questions, comments? If not, we can. No, yes.
Come on, no additional questions? You had one line about the registries for city. What do you mean? What do you expect?
I think it was Kimberly also was speaking about a CT scan in the United States, but also Richard, no? Yeah. Can you, would you wish, Rafael? No? It was, I think it was you, Kimberly, yeah. You were speaking about the CT scan registries. It's easy, not to get data in the United States.
I think this was regarding, well, first of all, we have to be careful about focusing on the right population, and we were thinking about pediatric. I'm not sure where that came up, but trying to find some way of capturing more low dose information and following the young children. I mean, the EpiCT data has really done that.
The idea is to make a collection of information about CT scans that are passed during pregnancy, so to identify the mothers and to be able to make a link with the potential health effects afterward in the children. Is it that?
I mean, there were, is Richard Wayford, there were general discussion of this point, but in terms of pregnant women, I think it is important to know what the CT scan is. So, for example, in the big Ontario cohort study that was mentioned, looking at cases of childhood cancer in women, a big birth cohort from Ontario, and identifying, linking it across to medical exposures during pregnancy. There weren't many, but I mean, it's obviously important under those circumstances to know what sort of medical exposure is involved. And if it's a CT scan, what sort of CT scan? Because as I recall, the CT scans that were involved with pregnant women there were of head CT. So quite low doses to the fetus. But if you don't have that sort of information, and I think maybe this is what this alludes to, you're really not going to be able to make any reliable estimate of the dose that would be received in utero. I mean, obviously, a head CT scan is going to provide a different fetal dose from an abdominal CT scan, for example. So it is important to have that sort of information if pregnant women are going to be are going to be exposed.
Okay, thank you. Just small organizational announcement. Somebody from Group A has left here in. No, so if it's this girl who has left or not. It's yours. Okay, so it's from Group B. Okay, so.
Okay, thanks a lot to both of you. And so if there is no more question, no. And so we move to Group C. So Cheyenne, you're going to make the presentation from remote.
Yep, I'll be doing the presentation on behalf of Group C. So thank you, everyone. As I mentioned, I'll be speaking on behalf of Group C. So our group was focused on looking at transgenerational and multi-generational effects on various biota. So we also had a question list, and we have divided up the questions into 3 different categories. So the first set of questions all looked at comparative approaches. So I won't go through all the questions individually, but in this set of questions, we focused on field versus lab and cross-species comparison, and are there ideal model organisms we should be studying? So when it comes to comparisons, whether it's between species or lab versus field studies, we kind of came to the conclusion that cross-species comparisons and lab versus field studies provide information that are more complementary than comparative. We think it's very difficult to really and precisely compare such differences, but we think it's more of a complementary information to get a better picture of what's happening. So lab studies allow for the control of experimental parameters, and in theory, you're limiting some of the confounding variables. But the advantage of field studies is now you're able to understand environmental relevance. Because as we all know, in the environment, radiation isn't really exposed in isolation. There's a lot of different stress factors that organisms are exposed to. One good thing of being able to conduct both field and lab studies is that by doing so, you're able to validate findings in the lab by testing them in the field, and vice versa. If you observe something in the environment, you can perform controlled experiments in the labs to verify those findings as well. So we think you need both because they give you complementary information. And when you're studying different species, it's important to recognize differences in radiosensitivity. So for example, something like a tardigrade is very resistant. It takes kilograms of dose before you are able to kill something like a tardigrade, versus mammals, which are relatively much more sensitive. So you have to keep in mind when you're comparing species, you may be talking about very drastically different levels of radiation resistance. And another important discussion that we focused on was differences in reproductive strategies between organisms. So this can include whether an organism undergoes clonal versus sexual reproduction, as well as differences in breeding strategies between organisms. So for example, something like a fish, which has a very large brood size, maybe a few hundred eggs, versus mammals that generally have much smaller litter sizes. So there's some different evolutionary and kind of adaptation questions we should be thinking as well. So these are kind of our discussion points. Moving into the next question of what is the best model organism, our conclusion was that there really is no single best model. What our discussions were focused on was identifying organisms that are most appropriate for testing of the specific hypothesis and questions. So when it comes to model or organisms available, we did want to highlight that very commonly studied models, things like C. elegans, zebrafish, Drosophila, Arabidopsis, to name a few, what their unique advantage is that due to very extensive study, so for example, the C. elegans genome is fully annotated, we're able to get very detailed information regarding ecotoxicology and environmental sensitivity. But we also think you can't only focus on these very commonly studied models. You have to also think about the less quote unquote famous species as well, because it's important to consider population and ecosystem interactions as well. So again, the more commonly used models give you some advantages from a study design perspective, but ultimately we think for environmental protection reasons, you have to consider populations and ecosystems. So that includes other species as well. And then we had a discussion about transgenerational effects in biota, in non-human biota versus humans. So one thing we did mention was that endpoints we consider for humans, so things like stochastic effects like cancer induction, they can differ from the endpoints we are more concerned about when it comes to biota. So for example, you may be looking at things like reproduction, growth, population survival. Within humans, we're generally considering individual levels of responses, but when we're talking about environmental radiation protection, at that point, we're considered more about populations and really whole ecosystems in the biota. Another thing that makes that's quite different between humans and the non-human biota is that human subjects are very intervariable. So just thinking about all the variation between human subjects of differences in lifestyle, mobility, access to medicine, and there's a huge list of these. You're introducing a number of potential confounding factors that could be interacting with the radiation exposure. So what we said was that we believe it's still important to consider transgenerational effects when designing radiation protection standards in humans, because we have evidence in other biota. So we think, although we don't know the full story, we at least should still be considering them from a radiation protection standpoint. And then the next block of questions were focused on experimental design. So here we were talking about different study designs, transgenerational effects in environmental risk assessment, dose rates, and things like that. So we ourselves in Group C came up with a few definitions, and I think this would be a great thing to discuss at the end of the presentation, because I think it is important to have consensus across the different groups. So how we define transgenerational exposure was you have an exposure of only the parent, so what we call quote unquote F0. And then what you're actually looking at, or you're studying the effects in a generation that was not directly exposed to the stressor itself. So what we kind of said was that none of the cells of that specific individual was ever exposed to radiation in this case. And since it's a transgenerational exposure, we kind of defined it that you're studying effects in F2 or later generations, but it really does depend on the specific study model. But our conclusion was similarly F2 or later. For multi-generational exposure, what we kind of agreed on was that it should be the same pattern of irradiation. There's different types of exposures. You can think of transient exposures, more seasonal exposures, or even chronic exposures. But the key is you have to keep the irradiation pattern the same across generations. And our distinguishing feature for multi-generational versus transgenerational was that for multi-generational, you cannot skip a generation. So every generation that you're considering was exposed to the stressor. So again, we'd welcome discussion on these definitions as well later. When it comes to study designs, we focused on kind of differences between molecular and omics style studies versus phenotypic and functional kind of outcome studies. I've listed all the points here. I don't have to go through them all, but what we said was that because there are differences in what you're studying, the types of questions and things you're answering, our conclusion was that it's important to consider different endpoints and levels of biological complexity. So in our mind, the strongest level of evidence is where you can show the progression from the initiating event, so subcellular to cellular changes, leading to eventually, hopefully, changes at either the tissue, whole organism, population, or ecosystem level. So our general conclusion was that the highest quality evidence would be studies where they can conclusively link, the initiating radiation exposure to functional changes in either the whole organism level or the population level. So that was kind of our discussion about study design. We also discussed a lot about dose and dose rate, which should we be focusing on. So we came up with a list of a few key study parameters that we should be considering when we look at these studies. So these include absorbed dose, dose rate, the time of exposure, so where in development and where in development the exposure occurred, as well as the duration, especially for these chronic protracted exposure, the total duration of exposure could play a role, as well as radiation type and quality. It's important to note that for non-human species, we don't have equivalent dose units, so we have to be considering differences in radiation type, quality, and things like that. And then we also mentioned that dose reconstruction across generations is important when studying generational effects, because you have to take into account consideration of historical dose as well. Because again, we're not just irradiating a single animal for a period of its life. We're talking about multiple generations that are being affected. So there's interesting ways you can consider quantifying total accumulative dose, things like that. So we want these are our discussion points for that. And then the last set of questions looked at transgenerational effects at the individual versus the population and the ecosystem. The image on the screen is just a very brief kind of summary of our model as we see it. We had a great presentation from Cisco on the effects and the role of epigenetics in evolution theory. So going into that, in terms of epigenetics and evolution theory, this is just one study we found, just to give you an example. But this study mentioned two things that we thought could be of relevance, including that could be of relevance for humans. So in this study, they mentioned that some genes are under selection to be imprinted, identifying the sex of the parent, leading to parent of origin dependent gene expression and effects. As well, epigenetic processes such as DNA methylation contribute directly to DNA sequence evolution because they can act as mutagens on one hand, but at the same time, they can modulate genome stability by keeping transposable elements in check. So ultimately, our discussion led us to believe that, yes, epigenetics likely does play a role in evolution, and we should keep that in mind when we're talking about multi-generational studies. And then we talked about ecological relevance of both trans and multi-generational effects. So one thing we focused on, especially when it comes to environmental studies, but even in the lab, it's important to realize that radiation exposure almost never occurs in isolation. So we should always be aware of abiotic factors, study conditions like temperature, pH, any of those kinds of factors, because there may be interactions occurring between stressors and there may be confounding factors in the analysis that you have to take into account. We also talked a lot about adaptation that has been demonstrated in animal models and the kind of general question of, is there a cost associated with adaptation to a stressor like radiation exposure? Another important thing we discussed was that in these studies, because they are being exposed chronically for long durations, one kind of study model you could look at is challenge radiation models. So you take a subject that was irradiated for multiple generations. If you then challenge it with a very high acute dose of radiation, do they respond differently than ones that were never exposed? Because if there are subtle changes in radiosensitivity, the hypothesis is that if they are in a slightly altered state of stress, maybe they respond differently to the radiation challenge. So that was part of the discussion as well. And that's it for what we had. I'm happy to open the floor to questions for myself or anyone else from Group C. Thank you, everyone.
So, some time for questions, comments, additions, corrections.
Do we want to start with the definitions, maybe, see if we're happy with those?
So if Rafi is okay with the definition, I think we don't have nothing more to add.
Yes, thank you. We had one question in Group D for you, because all the current investigation on future generation for biota, in which way it could influence the definition or the selection of the value for DCRL? Or do you have some specific sensitivity, or it should be for in which way we can expect some evolution in the future?
I can add a few words about that. We have already addressed some of these issues when we are reviewing the data for the report that will come in the next weeks. But for the moment, we cannot integrate them directly in the calculation of DCRL because we have to compare the sensitivity of generation F0 to generation F2 and be sure to have enough statistical power to do something about that. Because if you have only 3 papers, I don't know, perhaps we have more, but we need to have enough data for each RAP species in addition. So maybe it can be a sort of correction factor, or I don't know yet, but for example, this morning, from what I presented on Daphnia, microcrustacean, we could see that the significant dose rate from which we could observe an effect on reproduction began at the F0 at 35 milligram per hour, but then at the F3, it was to 70 microgram per hour. So it's something like that. It can change the threshold, and we have to work a lot during the coming 2 years to know what to do about that.
Okay, thank you. Additional questions? Questions from the online participants? So thank you very much, Cheyenne, for the presentation. I propose we move to the last group. So Group D.
So good afternoon. Today I and our colleague Freda Zolter, who is participating remotely, we are going to talk about the main points of our fruitful discussion from Group D regarding the effects of radiation exposure on offspring and next generations. First of all, I would like to talk a little bit about the equation of risk estimation. We have been discussed a lot about that, and it was our starting point. Very briefly, if you can see the equation, there are several parameters which include the baseline frequency, the doubling dose, mutation component, and potential recoverability correction factor. And for doubling dose, mutation component, and PRCF, the data is based on the results from mouse experimental model. And they use this data with the one assumption, but the mutation rate in human is same as in mouse. And for baseline frequency, they use the data from human studies. And if we go to the baseline frequency first, as you can see, the data based on the results for more than 40 years ago, and they take only Mendelian disease, chromosomal and multifactorial. Yeah, I forgot to tell that I'm going to talk about the preconceptional exposure, and Freda Zolter will continue about the postconceptional. Regarding the preconceptional, and based on the equilibrium and data which was used to make calculations, we formulated or our questions arose from the discussion of the questions which was given in advance. So first of all, regarding the baseline membrane, we take into account only Mendelian, chromosomal, and multifactorial. Should we include also cancer, and how it will change the results? And also, is there any data from human or animal experimental studies we can use to the parameters as mutation component on PRSC or doubling dose? Yes, next, regarding the generations, we have already discussed during the presentation of Group A, but we still have a questions. Like in the ICRP publication 60, they decided to limit the number of generation up to 10, but in ICRP 103, as in NANSCARE report 2001, we decided to use only 2 generations. Next, it's regarding the weighting severity. And here you can see that lethality was equal to 0.8 and quality of life with 0.82 in the years of life lost 20. And is there a differentiate according to type of disease? And will it not change but not contribute to the transparency or accessibility? So yes, this is the our questions regarding the preconceptional exposure. And now Fred Zolzer will continue with post-conceptional.
Yes, as you have seen, we have answered some of these questions by a counter question because we felt that if any of this is to be implemented in the system of radiological projection, more information is needed. And this is also the case with the questions for post-conceptional exposure. Part of them have been addressed already by the response of Group B. But I would like to present them here with the view of what would it change with the system? What would we have to take into account when we speak about those limits and regulations and so on? So one question is, are there non-threshold effects of radiation during the pre-implantation phase? That is, malformations induced during the pre-implantation phase, as it has been shown in some animal experiments. And what role does genetic predisposition play in this context? Is that something that could also happen in a human population, or is it limited to very specific mouse strains? If it is something that is can reasonably assumed also for human populations, then maybe we should have a dose limit, for instance, for women in reproductive age, which is not currently, as far as I understand, recommended by the International Commission, but is part of the national recommendations in some countries, such as Germany. They have a 2 millisievert milligray uterus dose per month limit for women in reproductive age. Then when we move on from pre-implantation to the organogenesis, how sure are we about the threshold doses for malformation, for instance, which we have only from animal experiments? Maybe we can have some, if we create a database for human not experiments, but experience, maybe we can add something to that. But at the moment, we only have information for threshold doses in animal experiments. And how sure are we about them? That's the question. How sure are we about the threshold dose for severe mental retardation, which comes from the atomic bomb survivor study? But it has also been mentioned in the presentation of Group B that if you look at the reduction of IQ, there doesn't seem to be such a clear threshold. So the question is, what should you look at this all or nothing effect of severe mental retardation, or at the IQ reduction? If we rely on animal data, what kind of safety margins would be prudent when we assume such threshold doses and we try to deduce recommendations for dose limits there? Probably we have to have some idea of how far below the threshold doses that we observe in animals should we stay? And then the question is, will there be any influence on dose limits due to the new data? For instance, what I mentioned, should we have a dose limit for, not for pregnant women, but for women that could become pregnant while working or while having such an exposure. Question 4 speaks about also post-conceptional exposures, the potential changes that could be proposed. And the question here is, should post-conceptional prenatal effects, for instance, cancer, be part of the detriment? And I think that has been addressed in, again, in the earlier discussion. Currently, cancer induced during pregnancy is not part of the detriment, but maybe it should become. Next slide, please. We had only this one question to Group C, which Thierry has already raised. Should the DCRLs be influenced? And yes, please go on. Question 6. What changes are expected in the classification and quantification of effects? That's a matter for task group 123, which is just starting its work. We noted that malformations can be can possibly be a stochastic effect when they come as a consequence of preconceptional or pre-implantation exposure, but they are a tissue reaction. They are considered a tissue reaction when they come at later exposures during organogenesis. So that is a question of classification of this and some other effects. Question 7, how to better consider the effects of in utero exposure in radiological protection in the medical field. Decision-making needs information about the risks, which we have, but of course we have great uncertainties, but we also need results. We need quantitative information for the possible benefits, and then we can start justification and compare the benefits with the risks. So we need robust justification of high-dose procedures in the radiotherapy of pregnant women, but we also need robust processes to avoid unwanted exposure of pregnant women in diagnostics or in professional exposures. Question 8, how to better consider ethical and societal aspects related to the effects of exposure to ionizing radiation in offspring and next generation. So again, both the pre-conceptional and post-conceptional. We discussed a couple of examples of where prudence is of particular importance here, because we are particularly unsure about some of the effects. We haven't seen, for instance, pre-conceptional induction of diseases in humans. We have no epidemiological evidence, but we have some animal evidence. So it is prudent to assume that it would happen. And the question is always how prudent should we be? Prudent is always a combination of precaution, be careful about what could happen, and also be reasonable, as low as reasonably achievable. So we don't want to reduce doses more than seems reasonable for us with a view on economic, societal and environmental effects, aspects. And then what we discussed a lot is how to include the stakeholders, how to show empathy to their concerns, even if it turns out that really all these effects only contribute a little bit to the detriment. But it is still important to very thoroughly consider them, because then we can say to the stakeholders, we have done our best to quantify all these different components, and we still have come up with this very little risk factor. So at least you can be sure that we have done our best to quantify it and not to hide anything or not to overlook anything. Justice is a question, it's a question of should for instance, male employees be treated differently from female employees, as they are to some extent. Justice is a question if it is the interest of the mother against the interest of the fetus. Of course, they are never completely separable, but there are many instances in this particular area where you have the benefits on the side of the mother, for instance, and the harm on the risk on the side of the fetus. So how do we compare these? Next slide, I think, just says it's the same thing. We just added here coming generations. Of course, that's also a question of justice, the question of intergenerational justice or intergenerational equity. We should behave in a way that we do justice to those coming after us. And the last slide just is a very general conclusion, as the other groups have also said. The impact on the radiation protection system can only be assessed after a new amount of evidence has been reviewed, and it is difficult to foresee the results of this review. So it is difficult to foresee at this point what it would mean for the system of radiological protection. Thank you.
Thank you, Frido. Thank you, Ilana. So questions, comments?
Thanks very much. Ooh, thanks very much. Chris Clement from ICRP. Nice to see you, Frido. I don't know if you can see me back, but it's good to see you.
Oh, can't see you.
Oh, it's for the best. Don't worry about it. So you made a very brief comment about the possibility of introducing margins of safety in the analysis, and I think we have to be very careful about where we do that in the system of protection. Obviously, margins of safety are important, but in my personal view, at least, it's not something that should be addressed during calculation of detriment. What we should be aiming for, in my view, is the best assessment of detriment with uncertainties on either side, because we know we can't do a precise job of it, and that introducing margins of safety should come later in the process, perhaps when we try to set dose limits or when we do optimization and justification. The danger of putting margins of safety in the calculation of detriment is that it's hidden, it's invisible, and it can be easily multiplied by adding more margins of safety over and over again in the system. So just a point to be careful about introducing margins of safety. Maybe it's necessary in some cases, but in my view, it should be avoided to the extent possible during the calculational phase for detriment. Thanks.
I see what you mean. And my comment was actually about dose restraints or dose limits. I didn't think in that respect, I didn't think of the detriment. And I don't think we discussed it in the group in that respect. But we do have, I think, a particular situation here in the things that I was addressing, in that we rely, in some instances, we rely completely on animal experiments. We have no significant human data. And that is why maybe margins of safety are more important for some of these aspects that we are discussing here than in other areas of radiological protection.
Thank you. Just one comment, just to emphasize that Frido is not a mentee of the task group, but Aidana is.
Just a matter of age. I'm too old for it.
Yeah, if I may, I fully agree with Frido. That's the only part in the calculation of the detriment, as far as I know, which is not coming from human results. So we have to be careful because many animal results are used outside of the radiation field with margins of safety. So if we have to consider that, and we have to clearly explain why we consider that finally it should not be there in our case, and to justify why it is needed for chemicals and not for radiation. So, or at least to discuss it and to keep it in mind. Yes.
Yes, thank you, Dominic. That's maybe coming back to my question of the first day, let's say the transferability of the animal studies to human risk estimates. So yeah, we understand over the 3 days that is difficult, but yeah, as you mentioned, it also is quite used in other fields like in medicine for disease and for drug discovery. That most of the compound are tested in animals and then used for human. So I think we have just to evolve in mentalities to try to be creative and in the risk estimates, let's say, equations to include this biological or animal cellular, let's say, analysis in a way to improve, let's say, the risk estimates. Thank you.
So same question as usual. Come on, questions, comments, criticisms, a point of final conclusion. Do everybody agree with this conclusion? I think it can be applied to group A, B, and C also, almost. Are there additional questions from people attending online? Okay, so if there is no more question, I propose we use the final minutes for the wrap up and what's next. So Cisco, I give you the floor.
Yes, about what's next. So the task group certainly will continue working on that, but before that, we plan to have a special issue in the scientific journal, and in the group A, we already identified some topics. So in minimum, I think that there should be a review of each of the groups, but I think that at least in group A, there is more information that can be divided a bit further. I think we can discuss this in more detail in the TG meeting, which is after the coffee break. Agree, but we do need all the experts and your contribution in this work, and so having reviews that have several authors, I think that we can focus on for each of them in the way that it's not too much work, but it's very complementary, let's say so. So yes, so I think that it would be very nice to get some feedback of the workshop. Of course, the conclusion is quite clear that we need to work on the materials and do some more reviewing, but did you find it useful? Sure. Yes. Yes. How did it work from the from the point of view of the remote participants? Could you follow this workshop?
Yes, we did. Sometimes, of course, there was a problem that some people reacted very spontaneously and didn't take the microphone, and so we didn't hear it online. But that was a minority of cases, and most of it was very well. We were well able to follow. Thank you.
Very good. That's nice to hear. And I think that also from our point of view, sitting here in Budapest, it was fairly easy to follow your talks, and I think this technical arrangement was very good. Anything else from remote? Oh, yeah, yes.
Yeah, maybe if I can say something, I thought it was very easy for me to follow it remotely, especially because I was part of Group C, which was mainly remote participants, so there was no problem of sharing the microphone around. The only thing that I regretted a bit was that I missed. But maybe that was my fault, that I missed the fact that we had to register for the IRPA conference in order to follow the presentations that were just given now between 11 and just before lunch. And therefore, I would have loved to have seen these, but now it was not possible, and that I really regretted a bit. But for the rest, I thought it was a very good workshop, and I was really happy that I was able to get it, follow it remotely.
Okay, and we also regret that it went that way. The idea was to really have a joint workshop. Okay, the slides will be anyway available, so don't worry.
That's nice. That's nice.
We'll distribute it.
If I may, about the slides, if we want it to be available to everybody, everybody has to provide the slides. So if it's the case already, that's fine, but I'm not sure it has been the case, especially for the ones that are presented just now. Yeah. So Cheyenne, for example, or if there have been changes, last minute changes, please send again the slides so we can collect everything. And in that case, the idea is that it is in a OneDrive, centralized in a OneDrive, which is accessible to everybody. So including the ones from the IRPA session. Yeah.
Who should we be sending slides to?
Aidana, I guess. Will you manage the slides to the Google Drive? Yes, she is our trusted mentee. Yes, please.
And also we ask to have access to this drive of mentees who are not having, because actually we wanted to use already some drive for work here and simultaneously, just like all together. And there was another wish from many participants. It's good to have kind of list, if nobody is against, to have all list like with names and emails, or maybe on the drive, or I don't know, somewhere, but it's also good for contact.
Thank you. I think there have been the list of participants and groups was distributed. And the emails were added later on, but they might have been in different emails, and it's not that easy to get. Aidana, can you explain? Yes, please explain about the Google Drive, how it works and all that, and what will be available there. Of course, we have the task group site as well, but we have more participants than people are there in the task group, ICRP page, I mean.
We have a OneDrive only for task group members. Should I create a new OneDrive for Budapest workshop? Okay. I have all your email addresses. I will create the OneDrive and to send you the access. So, and I will upload all your presentations so you can take it. Yes. And regarding the list of participants with the email addresses, if everybody is agree, I can also upload it there. Yes. Okay, sure.
So if somebody doesn't agree, please.
Yeah.
Nell, you had a comment?
No, just a thumbs up.
Okay, and I had a question about the agenda for the paper. So the idea is to have papers out of the workshop, but can you say a little bit more? The idea is to decide about different papers, to identify writers, and when is awaited the draft. Some approximate agenda.
Yeah, as soon as possible, I would say. Maybe, maybe who was keeping the notes for Group A on the points? I wonder if it was Fieke because he left. Do we have a list of the ideas that we wrote down in? Fieke has it. Yeah. Barakas, but Barakas is now the appointed. Can you show it? Yes. But anyway, here we have our guest editor.
Thank you. So when Cisco asked me a couple of weeks ago whether I can help with editing the special issue, as I'm one of the associate editors of IJRB, I haven't checked with Gail yet, but I am sure she would be supportive of having the special issue with IJRB. So in the morning, Group A discussed about having sort of reviews and papers for the proposed special issue, and we also discussed that we should come up with some sort of draft titles so that I can request Gail, what are the topics we are going to submit for the special issue. So yeah, and the authors. We haven't sorted out who would take responsibility for individual papers which we highlighted, but at least one or two of them we know who could be the potential contributors for the paper. For example, the first one, trio studies, and then the fourth one listed is congenital malformations, and probably both these papers will have some research data in addition to the review of literature. Okay, so that's what we discussed. And the other two, within the group, we will probably decide who would take responsibilities. And then we have also realized that some of us may not be experts in epigenetics. For example, we may ask someone from the other groups to join in writing these reviews or manuscripts, and hopefully if the other groups, maybe internally you discuss and come up with some contributions to the special issue. So of course there will be 4 articles or reviews for the 4 groups, and a summary of the workshop, editorial commentary or something like that. And then we also discussed in the morning that it's good to have at least between 8 to 10 papers for the special issue. That's probably a good number to handle within the time frame we have. And Andre, right, for Melody? Yeah, Andre suggested, and as Cisco mentioned, that usually the workshop special issue will have to be finished by 1 year after completion. So we hope that you all can contribute the manuscripts within the next 6 months, so that we will have enough time to review conducted and then published by within a year from today, for example. Is that enough?
And 6 months, I think, is a fairly good time. Yeah. Let's say by the time of the Vancouver, it would be nice to have some sort of copy already.
Yeah. For the other groups, is it okay to decide within your group so that how many manuscripts or contributions you can make? Is it okay? Yeah. Yeah.
Yeah, so we need working titles, let's say. It doesn't need to be final, but we need to create a plan to. Yes, okay. Excellent, thank you for this contribution. What else did we have?
For your information, there will be a presentation of the conclusion of the workshop that has been submitted to the European Radiation Protection Week. So for presentation, I'm pretty sure it will be accepted as a whole, I hope. And so I will come back to the rapporteurs or to have the feedback from the different groups. European Radiation Protection Week is the beginning of October. And another point is that for those of you that are not already members of the task group, there is a new position that has been opened in TG 121. So you have to look at the ICRP website. And so the position is to look for a geneticist, but if you are interested in participating in the work of the task group, let us know. And if you know some people that are precisely corresponding to what we are looking for, don't hesitate to tell them that it's very nice to work in an ICRP task group. Okay. So let's have a look at this call, and don't hesitate to circulate the information to people that may be interested, or maybe to participants of the workshop that may be interested in continuing the adventure with the task group in the next years.
Yes, Christopher, please come here.
Thanks, Cisco. Just since you mentioned all these great papers in Vancouver as well, the timing would be quite good if you would like to prepare. I invite you to prepare if you wish. Pre-recorded video summaries covering the topics of the papers and the topics of this workshop that can be part of the ICRP contribution to the ICRP symposium. We're asking all of our task groups to prepare presentations about their current work. And here, since you have lots of fertile ground, you're welcome to prepare more than one. It's no problem. Thank you.
Thank you. Okay. Breaking for coffee before the before the TG meeting. So I really wish to thank everybody, all contributors, TG and our esteemed guests for their contributions, and I hope we are much wiser now. I think so. Yes. Thank you.